So in part 1, we addressed the theory, the hypothesis, and the design. Part 2 is going to focus on methods, how we select populations and how we sample for quantitative research, how we collect and analyse data, and how then we can produce and disseminate our findings. So the next session is about the methods. How do you answer your questions? So just to take the baking analogy, for example, this is the how are you going to mix that cake to make the cake actually work effectively? How are you going to put your ingredients in what order, and what type of ingredients are you going to choose to make that cake reasonably decent? So the next 2 slides will just go over the different types of studies, because the methods that we use depend on what we're trying to do. Are we seeking to measure or analyse or correlate or compare or predict or infer? Are we going to have an experiment or a quasi-experiment, or observe or describe or infer? So it's those sorts of things that we really need to think about to make sure the methods actually fit with the research question, the design, and then the intended outcome. So one of the first questions that you have is choosing the populations that you're going to study or sample, choosing the subjects for your sample in a quantitative research paper. So there's a couple of different types of sampling techniques we can have. So probability sampling will offer some sort of stratified mechanism where you identify the population under study, and you'll just systematically identify who you're going to have, or look randomly to see to sample a certain number of populations. I'll go into how many people that we need to sample in the next few slides. The other type of sampling, which is used quite often, is non-probability sampling. So we quite often use convenience sampling. First of all, it reduces the amount of money that it costs to be able to get some data from populations and people, but it can also be there to be able to identify certain populations. So for example, if you only want to search for people with gastroesophageal cancer diagnosed within these types of years, aged between 70 and 90 years old, with certain characteristics, that's fair enough. You have to make sure that you absolutely do sample from a population which represents that question. Snowball sampling is something whereby you will get a number of populations, and you will ask those populations to snowball and ask their friends and their colleagues and their relatives and whoever else might be around who can also offer you some valid information which will answer your research question. All of these types of sampling techniques really do depend on the type and nature of your quantitative questions that you're asking. The most important thing for you to consider when you're reading your quantitative papers is, does the sample represent that question that those people are trying to answer? Would you have done anything different? If you sampled certain groups and you put them onto a control and an intervention arm, would those control people know that they weren't part of the intervention arm? Would that have affected their responses and their results? So you're the expert in the healthcare and the healthcare professional. So whether or not you would have done anything differently, would you have sampled a different population? Quite often you find very small single centre studies, which are great, and they do offer a significant amount of information on a particular subject. However, would that have been different in a different cultural context, in a different social context, in a different country, with a different healthcare system, which might offer a diagnosis more readily? Think about what it would mean in the context of where you are right now, and what that means in the real world. So in any quantitative methodology, we have to start to convert our responses into numbers so that we can calculate what that means in real world. There are a number of databases that we use to be able to store and retrieve data and perform some basic simple calculations. The one on the top there is SPSS, and that sort of shows you the types of responses and how we would start to put all of the variables and the cases within our data set. So the data set would consist of cases, or the subjects who are under study, and the variables that we're trying to identify. So there you can see rank, gender, athlete, height, weight, those sorts of things that are there as quantitative data. There are other more complex tools as well which enable a layering of data. So we can see on the bottom right hand corner there, this is a geographical information system. And what this does is it allows us to layer different types of data. So here we've got imagery, elevation, we've got all sorts of different things associated with addresses and boundaries and different features that are all geographic. And they allow us to layer that data onto patient-related data, but we're using a fixed point, something like a patient postcode or a patient personal identifier. The most important thing when you are considering storage and retrieval of data, we have to consider ethics. So you'll all be aware of the GPRD, the Data Protection Act, and a lot of other things that happen. But to be able to get data from our health services in the UK, certainly, you have to go through the National Health Registry services. So whether we have an integrated research administration system, we have to go through the usual sort of ethics frameworks, but we also have to go through NHS ethics frameworks on top of the university ones too. The most important thing is maintenance of safety of the patients at absolute most, and confidentiality as well. So when we are putting codes onto data and we are trying to apportion numbers to data and variables within our data sets, the one thing that we do have to think about is, the equality, the order, the intervals between those data numbers that we're using, whether or not there's an absolute zero, whether there's no zero. And there's a constant set of rules that we can apportion to sort of making these numbers work effectively. Nominal data are there to describe the things, to categorise the things. Things like gender apportioned to 1 male, 2 female. The numbers mean absolutely nothing, but they can be used. You can count how many number 1s you have, how many number 2s you have, and infer from that that you have this many males, this many females. All you can ever do with nominal data is add them up, present the number of cases, offer descriptive analyses to be able to describe what's there. Ordinal data, for example, is there to be able to give you sort of a bit more of a category, which, one person might describe their health as poor, one might describe their health as reasonable, one might describe their health as good, one might describe their health as excellent. There's some sort of relationship between those data. So you know that there's a bit of a line there, there's a Likert scale between feeling awful, feeling great. And that means something, so it means a little bit more. With ordinal data, so for example, then we can say how median range is, so what percentage of people felt okay. So a percentage of that would be meaningful to clinical practice. Interval data gives us even more meaning as well. So this one actually does have a 0 scale. So you're not going to have, it has no 0 scale. So what it does is it gives you categorized, ordered data. There are intervals between each and every score which mean the same. Body temperature, for example, distance, for example. They will give the reader an idea about what the differences is, not just necessarily between each number, but between each and every single number consecutively. Now, ratio data gives you that even more of specificity to this as well. So ratio data gives us categorized, ordered. There are intervals between the numbers which are meaningful. There is an absolute zero, and this is where height, weight, time, and this is probably the easiest one to sort of statistically analyse. These matter because dependent upon the type of data you have, depends on which type of data analysis you're going to perform. That data distribution really matters. It's not only about how your data are distributed, but how we start to look at how we measure and how we analyse the different levels of statistics. So we talked about nominal, ordinal and interval ratio data in the past, and how we start to apportion those and how we measure the differences between each one. This graph here offers all the different types of approaches that we do have with the different types of data. So we know that nominal data is just looking for differences in those data. So how many things are different between males and females, or between boys and girls, between dogs and cats, whatever you want to sort of identify. Ordinal is looking at correlations and differences between two different things. So for example, on a scale of 1 to 10, how uncomfortable were you, how anxious were you, and how does that correlate with, on a scale of 1 to 10, the Edinburgh Depression Scale, for example. Interval ratio data takes that correlation to a little bit more, and it gives you more sort of more tests that are a bit more developed and specific. They can start to test for absolute differences, different conditions. You can test for different effects of different variables as well. So this offers an overview of the different types of tests that you will see, and you'll see them in your quantitative research methods papers that you're trying to read. So the handy thing for you is just to look at what type of data are the researchers presenting, and did they apply the right tests. So have they done students' t-tests, binomial tests and chi-squares, for example, to describe their data effectively? Did they start to look at different sort of ANOVA tests to test for a number of different variables and the relations between those variables? So it's really nice to know this. You don't have to know absolutely everything about the different types of tests, but it just offers you a quick overview on what sort of things you will come up with and what names and tests that you will be expected to do on data when you start to become a quantitative researcher yourself. So in health sciences, we normally set a p-value at 0.05 as statistically significant. This can be contested, and there's a nice little paper that I've referenced at the bottom of this, which is also available in your Canvas module, which will just sort of talk to you a little bit more about that statistical significance and how we set it. However, I just wanted to identify what that really means in context. So the p-value is really that sort of level of significance within that hypothesis test that we talked about. And what it does is it represents that probability as to whether or not something has occurred because of chance or because of what you've put in place. So the p-value is used as an alternative to the rejection points, which provide the smallest level of significance at which the null hypothesis would be rejected. So what that means is low p-values, the less likely something by chance would have happened to have taken place. You put that onto your research question and your null hypothesis, a lower p-value means that something is more significant than a higher p-value. And I've written there how it corresponds to a sort of a percentage likelihood of something happening. And you can see there, if you're trying to test something to see whether it is or it is not going to be affecting something else through quantitative statistics, you want that likelihood of that thing to be taking place to be as low as possible. So p as 0.05 gives you that sort of parameter in healthcare research and sciences. And this is why it's important to understand the differences between mode, median and mean. In healthcare data, for example, a lot of our work doesn't present in that beautiful sort of bell-curved shape. So, for example, the negatively skewed distribution of data that you see on the left-hand side in this area, you'll probably notice for something like, I don't know, cancer diagnosis as age progresses, and you'd start to see cancer diagnoses of a certain types of cancer very, very minimum in the younger ages, and then it goes on and really increases as you get to sort of 60, 70, 80, and then tails off as you get to 90 and 100 because you don't have the populations. Distribution of data in the middle one, the normal one, would probably present you something like, I don't know, exam results, for example, where you'd expect to see a nice little bell curve there of some students who will reach around about 60%, other students who are reaching 100, other students on the other side who would be reaching sort of 40, 50, 30%. That gives you a nice normal, no skewed bell curve. On the right-hand side is a positively skewed event. So this could, for example, let's take this into a clinical context, it could represent something like childhood leukemia. So we look at this and the diagnosis and the age ranges tending to sort of towards the younger age groups. So again, you're looking at what that means in clinical context. What do the presentations of mean, median and mode mean in that clinical context? So let's go back to those elderly cancers. If we started saying that the modal value of cancers in gastroesophageal cancer, for example, was 68, the mode age, that doesn't necessarily reflect the whole of the data set. The mean, the average of diagnoses would be around about 56 to 60. The median would be a little bit between those two areas. So it's not giving us that clinical picture that we're really wanting. So it's really important to sort of not just identify mean, median and mode in your data, but to depict what that actually means in context. And sometimes what we do is we'll present the mean, median and mode, but also a standard definition. And a standard definition gives you that mean, average, but also gives you a plus and a minus of how far away the data are, the mean is from both data. So together, the mean and the standard deviation make up absolutely everything that you need to know about how your data are distributed. The 68, 95, 99 rule is based on the mean, but also with a standard deviation. So you can see there 68% of the population tends to be within one standard deviation of the mean. 95% of the population is within two standard deviations of the mean. And 99.73% of the population tends to be within two standard deviations of the mean. I think one of the most important parts of any research whatsoever is that dissemination of findings. It's the exciting bit. We've discovered something, we need to show people that we've discovered this. Now there's a great guy called Ben Goldacre, and I'd really advise you to look up some of his TED lectures. He does a lot of lectures on some of the data that has been found but hasn't been published in relation to pharmaceutical companies, for example, and efficacy studies in drugs. Dissemination of findings is absolutely essential. Many papers, many research journals don't publish null hypotheses, null findings. So if you have found something that affects something else, it's really important. People think that that's really important. But equally as important is if you haven't found something that affects something. We need to know something isn't effective just as much as we need to know that it is effective. Now the ways academics disseminate those findings are through peer-reviewed publications, and that's easy enough. You can access those through your library site and everything else. Through conference proceedings, so we often go to these academic conferences, nursing conferences, healthcare conferences that are associated with our specialities, and we can get a general idea about what other experts are thinking and feeling about certain clinical pathways and processes. There are also many other sources of quantitative data that we can take from. So there are many data repositories and data sites that we use who collect data on a routine and regular basis. The Office for National Statistics, for example, Cancer Research UK, for example, and a number of other NIHR, Joanna Briggs Institute, Cochrane database, systematic review processes, all there to be able to do a lot of that sort of presentation of quantitative data that you can access to be able to see if there's anything else going on. Just a quick note on peer-reviewed publications. Not everything you read is good. Not everything that you read will be of sound methodological rigour, and we have had a number of papers just recently that have been withdrawn because they've been published quite rapidly through COVID-19. Peer-reviewed publications, just check to make sure your journal is cited, it has a citation index. The reason for that is an impact factor as well. The reason you need a journal with a decent impact factor is so that that gives it that sort of valid and rigorous review. There are a number of predatory journals that have popped up in recent times which are there just to sort of get us academics to publish papers and pay to publish papers in them, but they're not necessarily following that rigorous peer review process that we try to make sure that happens throughout the whole of our academic careers. Okay, so this is just the sort of contents of a quantitative research paper that you can absolutely expect to be there. Every research paper starts with an abstract. Every research paper will give you a bit of a background which should really be based on a literature review. It'll present its methodology in the right way. It'll show you how it's sampled, how the collection of data happened, what procedures were taken, whether there were any pilots that were published in the past, what types of methods of data analytics have they done, and what sort of ethical processes have they followed. The results. When you're reading your results of any of your quantitative research papers, are they clear to you? Do the graphs make sense? Did they use the charts and the data and present them in a format that you understand? Are the results clinically relevant? Finally, you need to be looking at the discussion and recommendations, and the discussions should really draw from the findings of the research. Many papers go off on a tangent, they go off on a frolic and identify all sorts of different other things. Make sure that those results and discussions are related to the findings from the research study and are backed up with literature from news sources to identify what's going on in the wider research and the wider literature as well. The recommendations that your researchers will offer, are they related to the research? Are they offered by people who are known researchers in the field? Do the researchers have any integrity, credibility within the field that they're discussing? All these questions are things that you can ask. There's plenty of literature out there on how to critique a quantitative research paper, and I would absolutely advise just take 5 minutes time out just to quickly read one of those and go through one of your research papers and have a look. So what I tried to do was just to offer you sort of some information on quantitative research methodology, and I've done that by taking you through the processes and steps that a quantitative researcher would probably start to take. So from theory to hypothesis to design to methods, population sampling, data collection, analysis, and then the production and dissemination of findings. I've also sent you hyperlinks within this whole lecture to try and identify where you can go for further reading, but please feel free to contact me as well anytime you want to, ask questions. What I want to try and do is get you to realise that quantitative research and quantitative statistics are absolutely essential to any healthcare research. They offer a brilliant toolkit for us to be able to analyse our effectiveness, to be able to identify and predict what's best for our patients, and to give us a real good sound basis for our research and our clinical practice. So I hope you enjoyed it, and please feel free to contact me, and I've left my numbers in this final slide.